Lecture number 26, Transgenic plants developed for herbicide tolerance, pest and disease resistant, and abiotic stress resistant. Development of herbicide tolerant transgenic plant. Glyphosate and PPT are non-selective herbicide that kills the plant by targeting the vital metabolic pathways. So glyphosate is marketed as Roundup, and phosphonothricin or glufosinate is marketed as Liberty by Bayer and Basta by BASF. So herbicide tolerant transgenic plants are crops that have been genetically modified to withstand the effect of specific herbicide, allowing the farmers to control weed more effectively without harming the crop. So here the specific herbicide is for glyphosate resistance and phosphonothricin resistance. So herbicide tolerant engineered in crop plants by expressing the herbicide insensitive enzyme. For example, bacterial EPSPS enzyme insensitive to glyphosate is used to generate the herbicide tolerant plant. So second approach is the enzyme that detoxify the herbicide molecule is used. For example, the bacterial phosphonothricin acetyl transferase PAT enzyme detoxify herbicide phosphonothricin by acetylation. Engineering resistance to glyphosate. Herbicide glyphosate inhibits 5-enol pyruvyl cycumate 3-phosphate synthase, an enzyme of cyclic acid pathway in plant, and block the synthesis of aromatic amino acid. EPSPS gene is a nuclear encoded gene and enters the chloroplast using transit peptide. Glyphosate herbicide binds with more affinity than its normal substrate phosphoenol pyruvate and prevent formation of 5-enol pyruvate cycumate 3-phosphate. So here the cycumate 3-phosphate and phosphoenol pyruvate is combined by the EPSP synthase enzyme to form 5-enol pyruvate cycumate 3-phosphate. So this further converted into corisumate and from which the aromatic amino acids are synthesized. So the EPSP synthase enzyme is inhibited by the glyphosate, and this leads to this prevents the synthesis of aromatic amino acid and protein, leading to the death of the plant. Some bacteria contains EPSPS that is not sensitive or inhibited by glyphosate. So glyphosate resistant EPSPS gene isolated from Agrobacterium strain CP4. Expression of bacterial EPSPS in plant confers herbicide tolerance. Agrobacterium CP4 EPSPS gene expressed under CMV35S promoter. So since the bacterial EPSPS gene lacks chloroplast transit peptide, a chloroplast transit peptide coding DNA sequence taken from Rubisco was added at the start of the EPSP gene. So here you can see that bacterial EPSPS gene, before that chloroplast transit peptide sequence is added, and which directs it to this enzyme to the chloroplast. Plant transformed with EPSPS sp synthase gene of CP4 are tolerant to glyphosate. Glyphosate-tolerant transgenic plants are generally called as Roundup ready because it is able to withstand the herbicide Roundup. So the glyphosate sprayed on transgenic and wheat plant is shown here. So glyphosate inhibit the plant EPSP synthase enzyme but not the bacterial EPSP synthase. So because of this, the transgenic plants are able to survive due to the presence of the bacterial EPSP synthase enzyme. Whereas the wheat plant, the plant EPSP synthase enzyme is inhibited by the glyphosate, and due to this, the wheat plants die as it is unable to synthesize the aromatic amino acid. So herbicide tolerant soybean tolerant to Roundup is shown here. So all the wheat plants are killed due to the glyphosate spray. Here the soybean is able to tolerate the glyphosate. Crops such as soybean, corn and cotton are transformed with Agrobacterium EPSP synthase gene. These crops occupy very large area under cultivation. Mechanism of action of phosphinothrycin. Synthetic herbicide phosphinothrycin, also called as glufosinate, is the active ingredient in the herbicide marketed as Basta or Liberty. Basta. Natural herbicide precursor Bylofos, produced by the Streptomyces hygroscopicus, contain 2-alanine residue at plus glufosinate or PPT. Bylofos, due to the peptidase activity in plant cell, get converted into glufosinate. So here the 2-alanine residue are removed and the Bylofos is converted into phosphinothrycin in plant cell due to the peptidase activity. So glutamine synthase is an enzyme in the nitrogen assimilation pathway that utilizes excess plant nitrogen to synthesize glutamine. So the excess plant nitrogen is converted into glutamine with the help of glutamine synthetase enzyme. PPT or glufosinate, being a glutamic acid analog, binds to the active site of glutamine synthetase and inhibit glutamine synthesis. This causes ammonia accumulation to toxic level in plant and inhibition of photosynthesis leads to the death of the plant. So here the phosphinothrycin glutamic acid analog inhibits glutamine synthase enzyme. So because of this, the ammonia accumulates to the toxic level in the plant cell and kills the plant. Engineering crop plant tolerance to phosphinothrycin herbicide. Bylofos resistant gene encoding phosphinothrycin acetyltransferase enzyme detoxify phosphinothrycin by acetylation. The acetylated form of phosphinothrycin cannot inhibit glutamine synthase enzyme. Bar gene have been isolated from Streptomyces hygroscopicus and Streptomyces viridochromogens. So the transgenic plant expressing the bar gene or PPT resistant gene encodes phosphonothrysin acetyltransferase enzyme. The PAT enzyme convert the glyphosate sprayed on the transgenic plant to a acetylated form of glyphosate. The acetylated form of glyphosate is not able to inhibit glutamine synthase enzyme. Transgenic crop, corn, canola, cotton expressing the bar gene were produced by Bayer Crop Science and marketed as Liberty Link, as it is able to withstand the herbicide Liberty containing the phosphonothrysin. Development of transgenic plants for pest disease resistance and abiotic stress tolerance. Bacillus thuringiensis contains cry gene, popularly referred to as BT gene. The cry gene encodes for the crystal protein that possesses insecticidal, that is toxic to insect activity. During spore forming stage, crystal protein accumulates together as inclusion body as crystal structure. They are called as delta endotoxin. They are produced in inactive form and called as protoxin. So here the cry gene is encoded in the plasmid, and during sporulation the crystal protein accumulates, and this is also called as delta endotoxin. BT spores are used for the control of Lepidopteran pest. BT based insecticide are considered safe or non-toxic to mammals and non-target insects, and used for control of insect pest in crop field. But the main drawback of this BT spray is BT spores is stable for short time and loses its activity quickly due to exposure to UV rays. As it is applied on the external surface of plant, it does not penetrate the plant tissue, so that it is not effective for control of the borer pest. Different cry gene shows sequence similarity and variation in size. The size of the cry protein varies from 70 to 130 kilodalton. Cry1 group is effective for Lepidopteran, Cry2 is effective for Lepidopteran and Dipteran, and Cry3 group is effective against coleoptera pest. So the cry protein differ in size and exist among different subfamilies, but share a common active core comprising 3 domains. So here all the 3 cry group of protein have the similar 3 domains. So the cry1 group are inactive and are activated by the proteolytic removal of C-terminal sequence. So here the proteolytic removal of the C-terminal will activate the cry1 group of protein. Mechanism of BT crystal protein toxicity to insect. When the susceptible insect larvae feed on the plant sprayed with BT spores, crystalline protein in the spore dissolves in the mildly alkaline environment of insect midgut. So here you can see that when the crystal protein is taken by the insect larva, so this dissolved in the mildly alkaline environment of the midgut to form protoxin. So the protoxin is converted into active toxin by the activity of the insect protease enzyme present in the gut, cleaves at the end of the domain 3. So it cleaves at the end of domain 3 region of the crystal protein to release the active toxin. N-terminal region of toxin containing domain 1 and 2. Domain 3 at the C-terminal of cry protein have been implicated in the determination of the insect specificity. So the active toxin, cry toxin, binds to the midgut epithelial cell of the insect, and here you can see that domain 1 of the BT toxin binding to the receptor, and domain 3 gets inserted into the gut epithelial cell that leads to the um, damage to the epithelial cell. Transgenic plant expression of BT gene for insect control. Expression of full-length cry gene showed very low level of expression, so less than 0.001% of the leaf protein. Even the truncated cry gene showed slightly increased expression, 0.001%. So the analysis of the BT gene was done and it was found to be AT rich. This causes few problems. First is codon of BT genes are rare codon for plant. Second is AT rich sequence might act as a polyadenylation signal causing premature termination of transcription. Third problem is AT rich sequence acting as instability signal reducing the half-life of the mRNA. So to overcome this problem, increase the BT gene expression in plant to several fold. Usually a synthetic cry gene is made with altered sequence which has reduced AT content and increased plant specific codons. So this is the synthetic cry gene expressed under constitutive promoter CAMV. Accumulation of cry protein at high concentration in the cytoplasm of the plant cell can be toxic to plant. Also, the cry protein synthesis in the cytoplasm may be degraded by the proteolytic enzymes in the cytoplasm. For example, the synthetic cry gene is expressed, is nuclear transformed, and it is expressed. So in the cytoplasm, the high concentration of the cry protein level may be toxic to the cell. Second is the proteolytic enzymes of the plant may degrade the cry protein so that the level of cry protein is reduced. So to overcome this problem, the second approach to enhance cry protein level in plant is shown. This is by directing the cry protein to the chloroplast fusing the chloroplast transit peptide sequence, which increase expression by 10 to 20 fold. So compared to the first approach, here the expression is improved by 10 to 20 fold, and here for example the synthetic cry gene fused to the transit peptide at the N terminal is shown. So the cry protein expressed is directed to chloroplast where it accumulates at high level without any problem. About to problem. Third approach to increase the cry protein accumulation takes advantage of the prokaryotic origin of the expression machinery of the chloroplast through chloroplast transformation with the native cry gene. Okay, so this avoid the complications of AT richness of the BT gene. Furthermore, 50,000 copies of chloroplast genome per cell leads to a much higher copy number of introduced gene. was possible with organelle transformation compared to nuclear transformation. By expression level, BT expression level reached up to 3 to 5% of the soluble protein in the transgenic tobacco leaves. So the native cry gene is transformed into the chloroplast. So here the chloroplast genome, it is integrated, and this leads to the production accumulation of cry protein to a very high level in the plant cell. Commercially available BT insect resistant crop is shown. So cotton for the control of tobacco budworm and cotton bollworm is a serious pest in cotton. Genetically modified BT crops is shown. The Bollgard 1 cotton containing the Cry1AC and Bollgard 2 cotton expressing the Cry1AC plus Cry2AB. So both are resistant to cotton bollworm. So the BT cotton is resistant to cotton bollworm. So the second example is the yield guard expressing Cry3BB gene in maize. So these plants are resistant to corn rootworm. BT cotton expressing Cry gene. In 2002, India started BT cotton cultivation. BT cotton grown in 11.4 million hectare in India with a greater than 95% adoption rate. BT cotton containing Cry1AC, Bollgard 1, Cry1AC and Cry2AB, BT cotton is named as Bollgard 2. So BT technology helped India to triple its cotton production from 13 million bales in 2002 to 40 million bales in 2014. So the advantage of BT cotton and non-BT is shown here. In case of BT, small compact plant, many mature bolls ready for harvest, 3 to 4 spare for non-bollworm pest. So here the BT cotton field is shown. Many mature bolls are ready for harvest, compact plant size, and only 3 to 4 spray is required. In case of non-BT, it shows excessive vegetative growth, few bolls are ready for harvest and require 10 spray for control of all insect pests. Engineering plants resistance to pathogen. So in the state of Hawaii in USA, papaya cultivation was seriously affected in 1990 due to ring spot disease caused by papaya ring spot virus. PRSV infection in papaya, and in case of USA, Hawaii is a small island in USA where papaya is mainly grown, and this cultivation was affected in 1990. So to solve this problem, Dr. Gonsalves and his group developed transgenic papaya varieties expressing the coat protein gene of PRSV. So the coat protein gene of PRSV was taken from the PRSV Hawaii isolate and cloned under 35S promoter to use for papaya transformation. The PRSV coat protein expressed in transgenic papaya can potentially interfere with the uncoating of the incoming virus particle or somehow impact replication or movement of the virus. So the PRSV CP gene transformed into the papaya, and this will express coat protein. So the idea is the coat protein will interfere with the uncoating of the incoming virus. So this will the PRSV on entering, it will here the RNA genome. So this will bind to the coat protein so that here the uncoating of the incoming virus is affected to mediate coat protein mediated resistance. First transgenic virus resistant papaya was released for commercial cultivation in USA in 1998, and it was resistant to PRSV infection. Engineering plans for abiotic stress tolerance. Drought and salinity are the major abiotic stresses. Abiotic stress uh, also induce the expression of certain regulatory genes such as transcription factor that activate expression of abiotic stress tolerant gene. So in this regard, highly abiotic stress tolerant Arabidopsis thaliana DREB1A gene. So this is a transcription factor expressed in response to dehydration due to salt drought and cold stress was isolated, and the transgenic wheat with expressing the DREB gene driven by the salt inducible RD29 promoter showed enhanced resistance to water deficit condition. So here the RD29A gene and expressing the Arabidopsis DREB gene. So when it is expressed, the transcription factor binds to several genes and induce the expression of several stress tolerant genes.